Welcome to Biomed Unleashed.
Our podcast is endorsed by the MERLN Institute for Technology-Inspired Regenerative Medicine
and supervised by Professor Lorenzo Moroni.
We aim to make regenerative medicine accessible and understandable to everyone.
In our episodes, we invite scientists, specialists, and innovators to share their career insights and latest discoveries in this exciting field of regenerative medicine.
Martijn van Griensven is a professor at Maastricht University, located in the Netherlands.
He is the Vice Director of the MERLN Institute for Technology-Inspired Regenerative Medicine and the Chair of the Department for Cell Biology-Inspired Tissue Engineering.
Martijn's research combines cutting-edge science with a deep commitment to clinical translation.
His work spans musculoskeletal regeneration, biomaterials, and the development of innovative therapies that move from bench to clinical application. Hello, Professor Martijn van Griensven. We are very happy to see you today on our podcast.
Hello, Professor Martijn van Griensven.
Hi, very nice that you invited me. I'm happy to be here.
Thank you. So, to start, could you please tell us about your academic background?
Oh, that is an interesting one because it's a little bit different than other people, you know. So people normally study, do a postdoc, etc. So I studied in Leiden, and then I met a trauma surgeon from Germany during an internship that I did in the States, and he was telling, it's interesting that you have this biomedical sciences. We don't have that in Germany, and it's a kind of mixture. And he was telling, don't you want to come to us and work with us? And I was like, why not? So I went there, and I was thinking, cool, 1 year, 2 years, whatever, and that's it. And actually it turned out to be like 23 years in Germany and Austria. So it was a little bit longer than planned. And I was immediately actually jumped in setting up a lab, doing a PhD next by without really actually having a supervisor. And well, 3 years later I had a PhD, I had my lab, and yeah, things went crazy actually.
Wow. Yeah, because you started your professorship right after PhD,
Yes.
and you were also a first young professor in Germany, so that's all about it, right?
Yeah, I was 26.
Oh, wow. Wow, that's quite 7 more years for me. Yeah, not a lot. But were there any challenges with being actually so young and being already a professor?
Oh yeah, so it was really crazy because people see. So yeah, you have two things, right? So there's the one group that thinks that you know everything because you're so young professor, so you are, you know everything, which is absolutely not true, and it's still not true, right? I mean, I still learn every day. And then you have the other group that is absolutely envious, like, how is that possible that you are already professor? We needed to work so many years and go through so many steps and to go there. So they also try to block things for you, right? So they envy and then try also to block, so that make also a lot of hurdles coming in your way that make it sometimes also more difficult, right? So overall I'm happy that it's happened, and I'm happy how everything went, but there are some things that you say, well, would have been nice when that would not have happened, right?
So, following up, what initially sparked your interest in regenerative medicine?
Seeing the patients. So I saw patients in Germany, and they were really badly injured after accidents. So car accidents, motor accidents, and they came with like legs totally broken, arms broken, but really like devastated, right? So big holes in their bellies, right? So because they were hanging on a tree or whatever, and things like that. And well, you just saw that is not able to heal, right? It is just too big, that the normal healing processes that normally happen in your body can fix that, right? So I was thinking, well, that you need to do something, and so you need also to understand that it was end of the 90s, where it just was coming up, right? So I know you already had a podcast with Robert Langer, right? So yeah, and he was really one of the first ones that were doing that, right? So nobody really know how it was, but I saw these things. I saw this huge gaps. The body is not able to deal with it. You got amputations, I think, and things like that. And we were like, no, we need to do this regenerative medicine that is that is there, tissue engineering, that must also be applied for this, because these are the things that are prone to be used for that, right? So to really heal this terrible accidents and things like that, and that was like actually what sparked me to really go into that direction.
And you started also in Germany in Hanover Medical School?
Hanover Medical School,
okay.
yeah.
Okay. But then, so
because the trauma surgeon that asked me to come was working there, so that's why I started to go there.
Oh, so you were collaborating with someone and you started.
Yeah, I saw him during an internship in the States where he was doing a postdoc. Yeah, and he was from Hanover, and he was telling, well, that is super interesting. Yeah, don't you want to come working with us? And that's how it came.
Okay, but then you went also to the US, or you stayed in Germany? Or?
No, I was in the US as an internship. So during.
Oh, so you also went. Oh, okay. Yeah. Okay. Oh, that's interesting. Yeah. But then you ended up here in the Netherlands at the MERLN Institute for Regenerative Medicine.
Kind of back home, right? So I'm originally from Leiden. Yeah. So the Netherlands was the kind of back home again. Yeah?
And then, but why you came back? Why you came to MERLN?
So I was in Germany, and I was in Munich in that time, and I noticed I got it kind of stuck because I was working in a clinical department, and people started to get more and more like, okay, we need to do clinic, and that's where the money is. So research is like, less seen and got less money and was like more and more difficult to do. And then I was driving home in the train at that time, and I was seeing in ResearchGate actually an advertisement for the MERLN Institute for a department chair in regenerative medicine. And of course I knew the institute, it's also broadly known. I was like, oh, that's interesting, and it's in the Netherlands, which is my home country. It's exactly doing exactly what I do, regenerative medicine. Well, I spoke with my wife, and she said, "Oh, nice to go to the Netherlands." I mean, she is from Cuba, so she has no affinity with Germany or whatever. And she was like, "Well, the Netherlands is your country. Apply." And I was like, "Okay, let's apply. I perhaps don't have a chance, but let's apply." And I applied, and well, here I am. Yeah, so it worked out.
You like it here?
I like it here. Yes, I do.
Yes. So currently at MERLN Institute, your research focus has broadened to the regeneration of the entire body. Mm-hmm. However, as far as we know, you have also done a lot of research on musculoskeletal system regeneration, and particularly in bone healing. And if we talk about bone injuries, we know that there are situations where bone cannot heal on its own. And these large defects are usually called non-unions. Mm-hmm. Could you please tell us what are the main processes causing a bone not being able to regenerate, and why is it different for large bone defects compared for small ones?
Yeah, so indeed, because I was telling you, I come from the trauma surgery department, so therefore the musculoskeletal is one of the main things, right? So, and when you look at bone, a lot of people think always that bone is something that is dead, right? It's hard, it's calcium, and a lot of people think it's dead. It's not dead, right? In bone, there's a lot of fluid going on, a lot of vessels in there bringing blood for oxygen, nutrition, etc. And the big problem is that when you have a huge gap in your bone, that the ends don't see each other. So you need to imagine you're in the coast of the Netherlands, on the other side is England, but you don't see England, right? So if I put you in the water in the coast in Katwijk, for instance, you don't see the other side. So why you should start swimming to that side, right? Because you don't know you're going there. And that's exactly what happens in big bone defects, that they don't see each other. When you have a small bone defect, they see each other. So the cells see the other side, and they just swim to the other side and say, oh, there's a little gap, we need to fix it. But when you have a big gap, they don't see each other. So why should they go there, right? And that's why it does not heal when you have a large gap. Yeah? And that is also the problem that we need to tackle with our regenerative medicine, to make sure that the cells know that there's something on the other side and go there.
And what are the main ways to help to bridge this gap? And I know that some of them also focus on immune modulation, right? Because it's also very important for immunity to be activated to also help them go and heal the defect.
So there are two things. So one thing is that you want to have cells, bone cells in there that bridge this big gap, right? And how you do that is that you help them by putting something in what we call a scaffold, which is like a structure with holes where the cells can walk to the other side, so you make kind of a bridge there so that the cells know, oh, look here, and at the end they are in England, right? So that is one thing, and the other thing is indeed what you say, the immune system. So I think it's also a lot of time forgotten that the immune system is super important for healing, right? So when you have a hole somewhere in your skin or in your bone, you need your immune system, first of all, to keep it clean, to kill bacteria, viruses that are in there, to keep it clean. But on the other hand, what those immune cells are doing is also like kind of firing up your system. They are telling your bone cells, your stem cells that are in there, come on guys, we have a hole here, you need to be active, you need to work. So they give a lot of cytokines, how do we call them? So that are messenger molecules that are telling your other cells, there we have a problem, we are on fire, you need to grow. So they are really also kind of like the people with the whip, putting the other cells to work.
And out of all this ways to help the tissue to regenerate, what do you consider the most promising one, maybe in regenerative medicine? Going to more immune side, or maybe also vascularization, because vessels are also very important, right? Yeah,
you actually mentioned two of the main things. We need vessels because we need energy, we need oxygen, we need food, which is vessels are extremely important. We need the immune system to keep it clean and firing it up. And the third thing is what we need is really a filler, what I said, like the scaffold, like something that can bridge, right? So that are components that are super important. And then you need also something that tells your cells to be bone or to be cartilage or whatever you want to make. So you need vessels, you need the immune system, you need something to bridge, and you need something that tells, like I said, always a kind of like a poke-on, right? So that's something that feeds your cells to that they know what they should be, that they should be bone or whatever they should be. So it's not one thing. A lot of people always think, okay, you have one magic bullet. You don't have that. You need a combination, and you need a good combination.
Okay. So switching the topic up a bit. Currently you're investigating RNA-driven regenerative medicine. So let's start a bit with the basics. In the beginning of the 1990s, the first RNA-based therapies started to emerge. Could you touch upon what RNAs and RNA-based therapies are?
Mm-hmm. Yeah, so that is actually coming from very, very basic cell biology, right? So normally you have a gene, and this gene is telling the cell to produce something, namely a protein at the end. And the thing that he uses for that is RNA, which is in the middle. So the gene is making some RNA, and the RNA is then making sure that the protein is produced. So, and then the RNA disappears and the cell stops producing the protein. So our idea was like, okay, what we do a lot is actually using proteins, right? So not only we, a lot of therapies use protein, and what you say that the RNA therapy came because people were seeing, okay, it's very difficult sometimes to produce a protein that's correctly folded, it's the right size, etc., etc. So what if we take away this burden of making a protein outside the body somewhere to put it in the body? Why can't we use the body itself to make the protein? Because they are anyway good at that, right? The cell knows how to make a protein. So the only thing what we need to do is tell the cells which protein to make. And that's how RNA therapies came. So people start taking RNA, put it in. So that didn't come from a gene, but we just put in RNA, And the cell sees the RNA and says, oh, there's RNA, let's use it, and then starts making the protein you want. And that's how RNA therapies came. So you make just a protein that you want that the cell makes. And the advantage is that the RNA then disappears also. So you don't have a constant production, because often you don't want that. You want a production for a certain time and then it should stop. So therefore the RNA therapy was super good. So you use everything that is there in the cell, right? I always say it's like the coffee machine and it's making coffee, right? But you don't need all the other things. You just put the coffee machine in there and it's working.
And probably there are like several types of RNA-based therapies, right? Which ones are you focusing on in your research?
So the RNA-based therapy is really what do you want the cell to produce, right? So everybody knows it, for instance, from COVID, and they are produced like a part of the virus so that your immune system recognizes it. That's not what we want. We want that our cells know to become bone cells. And before I was telling you they need some pokon, right? Some plant food, that cells make you are going to be a nice plant. Exactly, that's what we do. So instead of adding the protein that tells the cells become a bone cell, we add RNA that makes this protein, and then the cells become bone cells. And the big advantages is that it produces in the right amount, it produces it also in the right size and folding, because they are actually very difficult to make. So the cells themselves just make it and stimulate themselves to become bone cells. So we use really bone stimulating RNAs.
And for example, probably also in bone regeneration, you have, for example, messenger RNA therapies, but also microRNA therapies, right? Can you please explain what is the difference?
Yeah, sure. So what I've been explaining before is messenger RNA. So that is the messaging which protein to make, right? So that is the very direct way. While we were busy investigating that, we also saw that the body itself has also some mechanisms of controlling that. So the body itself wants you to have a certain amount of protein, more or less, so they can regulate how long is the RNA, the messenger RNA, actually standing there. And that are microRNAs. And as the name already says, micro, they are very small. They are only a couple of centimeters long, right? Not more. And they target those messenger RNAs. So they go there and they make that they disappear. So meaning you don't produce anymore. So what we use that for is that we know that in certain bone diseases, when you have this big gap, you have some messenger RNAs that tell the bone, well, England is so far away, we don't go there, right? So they inhibit to build a boat that you go from the one side to the other side. And what we now found out is that blocking are microRNAs. So our idea is, okay, when we take out the blocker, the boat is being built and the movement is starting again. So that are in two different levels. So messenger RNA is really producing the protein, whereas the microRNA are normally the ones that block a lot of processes. And when we take out the block, the processes can run again.
And do you think any of these are better, or they're just different?
They are different. And they are different, and when you have a messenger RNA, you have always one protein. So you have one thing, which I think is also sometimes problematic, because the body itself never relies only on one thing, and that you see with microRNAs. MicroRNAs have several targets, so they have several messenger RNAs that they can bind to. So that also means that that the way a microRNA works is much broader. It's not as specific, but it's much broader. So therefore, it's also much more in the whole process. When you want to make the boat to go to England, you need wood, you need somebody that builds it, you need a sail, and there's, for instance, a microRNA that can inhibit the sail and the boat. So when you take out that microRNA, suddenly both things work again. Whereas with your messenger RNA, you only make the sail, or you only make the boat itself, right? So therefore, there are two different ways of using it.
Okay, and now if we talk a bit more about maybe off-target effects, because I guess then if microRNAs are targeting like everything, not everything, but more than messenger RNAs, then they have more off-target effects or consequences, or no?
Right, absolutely. That is again the problem with that, right? So that you then also have the risk that it blocks something that you actually do not want it to block, or that it blocks something that you want it, that keeps it blocking. And when you take it away, that something bad happens, right? And that's also what we are investigating at the moment. So how do these microRNAs look for their target? So what they do, is there something that we can do where it targets? And there you can see that it a lot of depends on how much is there from a certain target, for instance. So a microRNA on certain targets need a lot of target before it works, right? So if there's not so much, then it's fine. Sometimes the microRNA is in an environment where the target is not there. So then you also don't have to think about it. So what we're doing, we are also trying to see what is where. So where's the microRNA, where's the target, and even if it could be a target, but if they are not there, if the one is in that room and the microRNA is in that room, I don't care. It only gets dangerous when they are together, and then we need to think of how can we deal with that and how can we tackle that.
Okay.
And as you've mentioned before, another major challenge in regenerative medicine would be the immune system, that sometimes it can reject transplanted stem cells, treating them as foreign invaders. We understood that your research suggests that microRNAs inside extracellular vesicles, tiny particles released by cells, could help us control the immune system and reduce inflammation. Have you conducted any experiments or worked on clinical applications where microRNAs have successfully helped stem cells avoid immune rejection?
Yeah, we are not in the clinic yet, but we have done it in cell cultures. And we see that there are two things. So one thing is that these tiny bubbles, these extracellular vesicles, they are not complete cells. So it is not really recognized by the immune system. So that is one thing, that we can use them, we can bring them in without being recognized. So we evade kind of the immune system. And when you put in cells, that would immediately be recognized. The other thing is that we use microRNAs that actually target the over-exaggerated immune response. So when the immune system, I was telling you, that makes fire, that makes, it can also be too much. And when it is too much, then also your regeneration, so the healing process, is also blocked. So we also have microRNAs that is very interesting, that can block this over-exaggeration of your immune system. So and by that we use like particles that are not recognized, so the immune system stays quiet. Nevertheless, when it goes too much, we have a microRNA that can actually dampen this fire situation.
So you said, and this is not in clinical trials yet?
No, it's really still very basic research, where we are looking into mechanisms, how is it working, and then if we know more, then of course we would go to more in vivo, so more animal trials, and then finally go to the patient.
And is it the same for all microRNA treatments, or?
Very much, but there are already microRNA treatments that are in the clinic. So for instance, in certain liver diseases, there is already clinical trials going on where they use microRNAs to dampen the immune response in the liver, and thereby rescuing the liver. So we know that per se using a microRNA in a human body, is not dangerous, right? So that also gives us really the strength and the will to continue this research also for our systems, for our bone, for instance, or for the immune reaction that we see. to use it there.
And do you think it will be different for different microRNAs, like when we're going to use them in human body? Or because you said we already used one type and we know that it's safe, but do you think it's going to maybe completely different for another microRNA type?
So I think per se, the use of microRNA, so the molecule, I mean they are very similar. They are built of similar Lego parts, right? So that part we know is not dangerous. For each microRNA, what you need to see is which targets do they have, because each microRNA has different targets, which can be also lead to adverse reactions. So for each microRNA, even if we know that per se bringing it in the human body, it is not dangerous, we still need to check for each microRNA if it's dangerous because of the targets they have, right? Because when they have then one of the targets is essential for living, then we need to really think, can we use it or not? But per se, we can use it, but for each microRNA still you have to check, if it's really safe because of their working mechanism they have.
As far as we understand, cells respond not only to chemical signals, but also to their physical environment. Things like pressure, stiffness, mechanical forces can all influence the behavior. So we were wondering, have you studied how these environmental factors could affect microRNA expression in stem cells?
Oh yeah, absolutely. So we know, for instance, when you look at bone, I'm standing here with my 85 kilos on my bone. That means my cells get constantly some pressure, right? And that is good, because that keeps them fit and they know I need to make a strong bone, right? That's also what we see in patients when they have like a fracture. We want to have them as fast as possible go walking again. Physiotherapy, super important, because when you're just lying in bed, the cells are like, okay, why should I make a bone? Because there is no force, right? So you need to stand on it so that your cells, notice, oh, I need to build a little house around me because I need to be protected, right? So, and what we have seen is that indeed with certain mechanical stresses, so load, certain stretching, that we get different microRNAs. And actually, one of my postdocs is now starting a project together with the ETH in Zurich, where we are looking at stretching, and in this case it's tendon cells, stretching them. We know the mechanical properties. And then looking at which microRNAs are then produced and coming. And we do see indeed differences depending on the stress. When you have a normal stress, it's fine. When you have an overstretching, you get completely different microRNAs as a kind of protection mechanism probably. So we see that indeed also mechanical environment is influencing what microRNAs you have.
And since you said that you're only on the basic science stage, right, in the research?
With the microRNAs?
Yeah, with the microRNAs, I mean. Did you use already like a 3D environment for your research, or is only in 2D? Because as far as we know, there is also like the difference, right? Because in our body, everything is 3D, but we can also culture, for example, cells on 2D, and it's going to be completely different.
Yeah, absolutely.
So have you tried already with 3D culture?
That is one of our main things. So we do something in 2D, right? So just to have to start, but then we always go to 3D, because that is super important. And we have translated actually to the clinic like 3D printed scaffolds for bone healing, and those we take actually in a little bit smaller version, so not a 20 centimeter big thing, right, but a smaller version like half a centimeter by half a centimeter in the cell culture, to give the cells like a 3D environment. So we really mimic what we do also in the clinic. And what we also do is that we use like so-called spheroids. So we take a lot of cells and we let them make a little ball. So we let them make a little round bone like type of thing. That is 3D. Where the cells see a cell above, below, left and right. And they speak with each other, like in the body. In the body they are not next to each other, right? The bone cells are not next to each other. So they are really like in how it is in real life, 3D with above, below, etc., and a little bit apart, because bone cells are never lying very close together. When you do it in 2D, they are next to each other, but in the body they are apart. And in this spheroid, they are also apart. So that is much more like how it is in the human body. And that's what we try always to do, to really mimic as good as possible the situation how it is in the human body.
We also know that there's a microfluidic cell culture system, which are tiny devices the size of a few millimeters that can allow scientists to precisely control cell environments. Have you used these systems to study microRNA activity, for example?
Yes. So again, that is a little bit similar to like how we use these little spheroids, right? Now also in the microfluidics, what we can really also do there is like, as I said before, in the bone is not just a piece of calcium, right? It is really something that is living. There's blood going in, but there's also fluid that is moving up and down, and that is exactly what you can then mimic also in your microfluidic system. You can have very tiny fluid movements there, but exactly that what you also use, what you know that is actually also in the body happening. So we have also used that once, and we have very good people in our institute that can build such systems. And when I say I want them like with this form or this form, they can make it. And we can really mimic the bone situation, how it is normal. But the beauty is also we can also make it how it is like in a traumatic environment. So we can make there in this microfluidic system as if there is a fracture. And then see how microfluidics play a role. Do they change? Can I do something? And of course, then we take the cells out, we take the fluid out, and we investigate the microRNAs and looking for, okay, we did something. How did that change my microRNAs?
And for example, so we talked a lot about the environment of the cells and the microRNAs that are inside the cells. But have you studied any like specific materials, for example, for bone regeneration that may somehow influence microRNA expression or something in the cell?
That's what we're actually doing right now. So we are looking right now to different biomaterials and looking how do the cells respond to that, because the cells are actually the ones that are producing the microRNAs. So we are now looking like, okay, the material itself, so the chemical composition, does it change? And we did a very small study with a group from another department that had a very nice two different chemical compositions, and we saw that when we put cells on, one getting more into bone and the other not so much, and we could actually see that the microRNA composition was completely different. And the one for the bone forming was indeed getting microRNAs that we know are important for bone formation. So we there see also that per se the material indeed is also super important for what microRNA do you get. And the second thing is, it's not only the material, it's also how the material looks like. So how is the texture of the material? Is the material with big bumps? Is it with little pillars? How is the distance there? So what is the shape of your material? And so the founder of the institute, Clemens van Blitterswijk, he actually saw that that forms instruct cells. And we now can actually see that indeed the form instructs the cells, but also to make a different microRNA. So it's not only the material, it's also how the material looks like. So how is the shape of the material lets you also tell what type of microRNAs are produced actually.
And in relation to microRNAs, so we know that there's this thing called Antagomirs, which are synthetic molecules that can block microRNAs activity. Can you explain how these Antagomirs are used in regenerative medicine?
So what we see is that I told you these microRNAs are coming and they block a messenger RNA, right? So we know, for instance, that in certain diseases, there's way too much microRNA. So they block actually good messenger RNAs, and therefore you're not healing your bone, for instance. So the idea is, okay, this microRNA that is coming here and blocking my good messenger RNA, I want to get rid of it. So how to get rid of it? Well, a microRNA is a sequence, and this sequence is has a copy. So you can make an exact copy of it that exactly fits to the other side of the microRNA. So instead of the messenger RNA, you can make something that exactly fits there. And that is your antagomir. Yeah? Antago is against, yeah? So against the MER, against the microRNA. So you make an antagomir that is exactly a copy of your microRNA that blocks it so that your microRNA cannot come to the messenger RNA. Yeah, and thereby you make sure that your microRNA is gone. Your messenger RNA is still happy, yeah, and starts doing its job. And thereby you do better. And that we have indeed used. So we have looked at patients with osteoporosis, where we saw microRNAs that were highly upregulated. And so we made antagomirs for those bad microRNAs. And we looked first in cell culture, and we saw indeed the cells were much more happy, they were producing much more mineral, they were making bone. And now we have done actually that in an in vivo study. So we have used a model where rats got osteoporotic, had a fracture, and there we injected the antagomirs. And what we saw is that the bone healing was much faster and much better. We got good hard bone, and normally in osteoporosis it's a bit brittle. And there we could see it's really nice bone, hard bone, and it's working. Yeah? So we are currently actually trying to get this to a higher level and bringing that to the first inpatient study.
And what challenges can come with using these antagomirs?
A little bit the same as what we spoke about with the microRNAs, because the microRNAs have different targets. Yeah, it can also be that when you use this antagomir, it's an exact copy for your microRNA, but maybe there are other microRNAs that are a little bit similar. Yeah, only in one or two spots they are different, and your antagomir can still block it. So it can be that you can block the wrong microRNA. So you can block a good microRNA, and that's not what you want. So we are really at the moment looking at the specificity. So how good is your antagomir actually in recognizing the microRNA that we don't want? Or is it also doing others? And can we then inhibit that in a way? So that is one challenge we have. The other challenge we have is like, can we get enough in there? Yeah. So how do we bring those antagomirs in the body? Do we do it local because we want to have it in the bone, or we don't want to have it in the skin, or what we want to regenerate? Can we bring it local? And how much do we need? And how long does it need to be there? Because the cells are producing the microRNAs, right? So when you block some microRNAs and new ones are produced, do I still have enough antagomir or not? So that are challenges we are currently investigating. So can we have some system that gives antagomir over time? So not everything at once, but a little bit, every time a little bit, so that it's really every time a new microRNA is coming, it's blocking, it's blocking, it's blocking. And how do I get it there? Can I inject it in the systemic, or do I need to really have it on the spot where I want to have it?
But also you taught that microRNAs are not very specific because they target different different molecules. But then, for example, if you use an antagomer that targets one microRNA that may have influence on different things, and maybe they're not bad things per se,
Sure.
and then if you block it, so it can not only block a bad thing but also a good thing, or...
Exactly, that is one of the challenges, right? So, and that's why we also look at the microRNAs. For instance, we found for osteoporosis, we found one microRNA that indeed has several targets.
Okay.
But all of those targets, we want that they work. Yeah, so that was a lot of work. So we really researched 10 years to find this specific microRNA that has really everything it blocks is actually what we need. We found also others that blocked things that we need, but also blocked things that we indeed don't need, right? So that one we didn't want. So it was a lot of work to find exactly this microRNA that has such a spectrum, that we really say, well, if we take it out, everything good is coming again. So that was bad. That was really a lot of work, and we also see that now blocking that one with an antagomir indeed brings all the good things up again. So it was a bit of luck. But at the end, we found one that was indeed not having this problem of like, but also some good things are now not working, right?
So going back, you've explained some of the challenges in using microRNAs for therapy would be, for example, to deliver them to the target cells. Scientists have developed different delivery methods, like including viral vectors, so using harmless viruses to insert microRNAs into cells, and I think also lipid nanoparticles, so fat-based carriers that protect microRNAs as they travel, as well as, for example, biomaterials, so the engineered structures that release microRNAs slowly over time. What delivery systems have you explored in your research, and which one would be your favorite?
So we have explored the last two that you mentioned. So we have looked at these fat little bubbles, yeah. Because the nice thing is, because they are from fat, they can really melt with the cell membrane, which is also fat, right? So that automatically goes in, it just melts, and the microRNA is in your cell. So that is super nice. And we have also used the biomaterials. So because what I told you before is we want to have this bridge, for instance, in the bone, this scaffold. Yeah, and when there the microRNA is on, yeah, what we envision is that the cell is starting to walk. Yeah, and in the moment it hits your microRNA or your Antagomir or whatever, it gets in, right? So we have done both things, and even we have tried to combine them, so that we made a microRNA in a fat bubble, yeah, put it on a scaffold, yeah, so that we have it in place, but have also a good modality to really get it easier into the cell. And I really think my favorite are the fat bubbles, because they really make very sure that my microRNA is really getting into the cell. And we are now working together with a company actually to try that we can make it even more specific. That we say when a bone cell comes by, you go in. When some other cell goes by, don't bother to go in. Yeah, so we do that with specific door handles, yeah, that we try to put on these fat bubbles, yeah, that only work for a certain lock. Yeah, and when a certain cell comes by, the lock doesn't fit. It goes by. When the bone cell comes, it locks in and it melts together.
Okay, and if we think about combining microRNA technology and, for example, other innovations such as CRISPR, the gene editing tool, do you know, for example, if it's possible to modify these microRNAs using these gene editing tools, for example, CRISPR?
So we ourselves have not looked into that. I know of some groups that indeed look into that, because you need to imagine that the microRNAs are like messenger RNAs, also produced from the genes, right? And when there's something wrong, that too much microRNA is produced, you can also imagine that indeed with CRISPR-Cas you can say, okay, I can cut a part of the microRNA out, or I can cut out some of the elements that makes that it is produced too much, right? So people are indeed looking into that, how to engineer, yeah, indeed to get the right amount of microRNA. Yeah, you always need some microRNA, right? But the right amount. We ourselves are not working on that, but some people are looking into that, and it seems indeed that it's also a way of perhaps changing the diseases.
And what potential advantages and challenges could this approach have in general?
When you use CRISPR-Cas, so the advantage would be that you can fix it forever, right? So, or kind of forever, like, because you really do it in the cell where they are produced, yeah. Whereas what we do is we let it produce, but then take it away, right? That is an advantage. On the other hand, the disadvantage is, of course, you take out something in the cell, and thereby you change your cell. And when actually that is perhaps not so good, you have your changed cell, and it is then very hard to revert it again.
Okay, and if we talk again in general about regenerative medicine, looking ahead, how do you see regenerative medicine in 5 to 10 years?
So I think the way we have gone into now is really interdisciplinary. Yeah. So before it was people looking at cells, people looking at materials, scaffolds, people looking at these stimulating molecules, and now what you see more and more is that people really start combining that. Yeah. So you have chemists, you have biologists, you have all kinds of specialties that really work together. So I see in the regenerative medicine in 5 to 10 years that it's really an interdisciplinary field that really tries to combine the best of each of the parts. Yeah? And I think that also will be then getting to a more successful story, right? Yeah, because I think what we have been speaking now, how do I get my microRNA there, right? And we need the material, we need chemistry, yeah? So we need also the cells, right? So that take it up. So I think that is what is really going to, and I think that has really started and with this interdisciplinarity, and that is also what will bring the field forward, I think.
And which countries are leading in regenerative medicine research?
I think the States they have very good groups. I know also that in China there's a lot of effort in there, but it's sometimes hard to capture because they don't not always publish in English-speaking journals. And in Europe, I also think that, for instance, the Netherlands is certainly not bad. Not because I'm Dutch and tell that, but we really try here also to bring it much faster to the clinic. I've worked in other countries and where I see a lot of keeps hanging before the clinic because there's a lot of hurdles put up to test it in the clinic. And here it's really an open-minded country, and when you have really enough safety data, etc., then you say, okay, let's really bring it to the patient because that's why we do it for. And I think that is what is happening here. What we also did, we have treated patients with 3D printed scaffolds, with cells, with stimulating molecules, and we saw it was helping them, right? So, and I think that is, that is something that is extremely, important to do, and I think that's also why I'm so happy to be here in this institute, because we really can be translational and bring the newest techniques really to the patient.
And here in your work at MERLN, are there any upcoming projects or something that you're particularly excited about?
So I'm on the one hand excited about my project where we really are working in patients with non-unions, what we spoke about before, and really treat them together with the trauma surgery department in the Maastricht hospital. So that I find fantastic. I have also a colleague that is working on together with ophthalmology, and she is also going into clinical trials for patients that are almost blind because the cornea, so the front part of your eye, is troubled. And she has also made regenerative medicine therapies that she is now also going to try in the clinic, and I think that's really fantastic to also for that patients, that type of patients, because they are almost blind, they can hardly see, and when you can really with such a therapy can have them look again well, even if they may need some glasses, but they can really see again. I think that's fantastic. So I'm really happy about that, and that's also so nice to see it really goes to the clinic, and that's also so nice here that we're here in this building, and over there we have the clinic. It's one bridge further that you have really this contact, and you don't need to get in your car and drive to the other side of the city, for instance.
In talking about your projects, what is one of your research findings or projects that you're most proud of?
Yeah, that is really the bone engineering part. So I've been really working there 15 years in trying to get the scaffold and the cells right and the growth factor right. And when I came here, there was the surgeon Martijn that was then really say, well, you have been working on that, I see your results, it looks good, let's put it in the patient, right? And that was such a nice feeling that okay, I've been working all these years on it in the lab and doing all those things, and now it gets into the patient, that was a wonderful feeling. I was just 3 months here in Maastricht and we did that, and that gave me such a boost to see really you've been working on that, and people were asking in the lab, and is it really going to bring something? And then you see, yes, it brought something, and that made me very, very happy. Of course, I have other projects that I'm also very excited about, etc. But this is something that was really, a highlight.
And do you have a specific example, like maybe like of a patient or a situation when you used your scaffold, your tissue, right, that you made, and maybe like an example?
Yeah, so there was one patient, actually it was the first patient that had a motorcycle accident and had like a 15 to 20 centimeter gap in his shin bone. Was huge. It's big, yeah, and that's exactly what happened. You know, it was not seeing the other side, and at the end it was like, okay, we amputate. He's going to be amputated, yeah, which is not nice. And then we came and said, well, we may try this, and we did it. And then I saw him 1 year later, and he's walking without pain, and he's walking up and down the floor here in the MEMC. He is happy and say, okay, you know, I'm going on my motorcycle again, and I'm happy, right? And that is just fantastic, you know, to see that he has both legs walking like normal, and yeah, no pain, no nothing, and yeah,
Wow.
just living again. And that is nice, right? So it's not always working, unfortunately, but when it works and you see such a patient, you know, there you get really, excited and emotional that you see, wow, yeah, that is nice. Yeah.
And for example, you talked about this very, very large injury, but is there like a specific size or distance when the body's like, I'm not healing this, I don't know what to do?
Yeah, so there people think about two things. So one thing is like when you have the diameter of your bone, and when you have a gap that is 2.5 times the diameter, people say it cannot heal anymore. So because that's too far away. Or people also typically say, well, up to 5 centimeters, it can heal, right? But as soon as it is bigger than 5 centimeters, actually the chances are very low that you can heal it by yourself.
And if we talk about your work process, there is a AI, artificial intelligence, that is everywhere right now. How do you feel about it? Do you use it, or you're more skeptical about it?
In the beginning, I was certainly a bit skeptical because it's also like it only knows what you have fed to it, right? At the moment, it gets more and more. You know, you see also it gets more and more scientific, right? In the beginning, it was also not scientific. And we have used it a little bit to look at the microRNA and its targets, you know, because there are so many databases. So we were afraid that we may miss some of the targets that may have microRNA. So we have used AI to look into that and see, okay, this microRNA, which targets do you find? And then he comes up with all the targets that he can find actually, and that is really helpful for us. And then we always check it. And we still check, is the source okay? Is it indeed a real target, or is it really in 0.001%? Okay, we don't have to think about it, but that is something that we indeed can use for. Yeah. Another example is also that people have used it also for looking at the scaffolds. Yeah? What is the best scaffold structure? Is it round holes? Is it other holes? We have done something, but with AI you can also try helping to understand better how can I build my scaffold perhaps in a better way, more natural for the cells.
And for example, if we talk about personalized medicine, right? So you want to make products that are personalized to specific patient, because different patients have different problems, different defects. How do you see AI playing a role in that as well?
I think that it can also play a role because what we also envision is that we make like a big array, so a big screen of microRNAs, for instance. Yeah, so you get like 300, 400 microRNAs that are different in this patient, and to be able to really capture that for yourself and to bring that in conjunction with, for instance, stem cells from the patient and this that. You have millions of possibilities there, right? And I think with AI you can much better evaluate those whole data set and then start looking into, okay, what are really the microRNAs that play a role for this patient, and then make it much more personalized in that sense. It's still future, but I can envision that for that we could use it.
And continue talking a bit about future. What type of skills do you think students need to possess for regenerative medicine?
So they need to be open-minded, first of all. And I think they need to be able to understand the different aspects. What I said here, you have people that have chemistry with biologists, you have cell biologists, you have people that doing the gene therapy or the proteins for stimulating the cells. So I think when you are a regenerative medicine student, you need to have like a basic understanding of everything. You need to know a little bit. Okay, what is a scaffold? You don't have to be the hardcore chemist, right? Because therefore we have the hardcore chemist, but you need to understand a little bit about the chemistry, about the material. You need to know about the cells a bit. What can they do? How can I use them? What can I do? And what about the stimulation factors? And that you are the one that is the director, right? That can then handle your regenerative medicine, and you bring the things together. And you are the one that knows. I know this super chemist, and what he makes may be super interesting because the surface he makes, the cells would be interested in. Bring it together, right? So I think that is really what it is about, so that you understand the entire field and not very narrow. So I think you are really a broader person, a broader scientist in that sense.
Okay, and our final question for today. Actually, the time went so fast and we don't want to finish it, but it's actually maybe a bit surprising. But what would you do if not regenerative medicine?
Ooh, what would I do? What would I do not regenerative medicine? I think then I would go into infectious diseases. Because that is also a very big problem in the world, right? So with infections and bacteria that are resistant against antibiotics, etc. So I think that would be then a field that I would then more going into microbiology, infections, things like that. I think that would also be something. And when it would not be medicine, then actually I think I would like to be a musician.
Oh, wow, what music?
A guitar. I play guitar, and would be something that I would then like trying to make a living out of it. But I'm not good enough now for that. But that would be something that I would then go in that direction.
Okay, perfect. Thank you very much, Martijn. And I hope you liked it. At least we liked it very much.
I liked it very much. It's very nice.
Yes, thank you very much.
Thank you to both of you.
And also I would like to introduce you our team. Come here. That worked a lot on making this video, this episode for you. So thank you very much. Amalia Bukdesaran, Misha Bagirov, Alba Viagrasa Martin, and Elisa Avsyanika, and our guest Martijn van Griensven. Thank you very much.
Thank you.
Thank you very much for listening. We hope you enjoyed this episode of Biomed Unleashed podcast and will stay with us for the next ones.